our first speaker, and it's Hannes Bretschneider. Do I say it correctly? Great. From Deep Genomics for conversation. And I will unmute you as well.
Yeah, so thanks a lot for the introduction. So I'm going to talk about this startup that I co-founded about 2.5 years ago together with my PhD supervisor and some other people from the lab. So we were at the University of Toronto, and my PhD supervisor was Brendan Frey. And we were doing research on using machine learning in biology. We founded this company in 2015. Has literally been a case where, well, we're throwing ideas around over lunch, and it just came up, oh, why don't we start a company? And by now it has ballooned to a company with 30 employees. We've had 2 venture capital funding rounds, raised in total $50 million. So it's been pretty astonishing. However, a month ago I actually decided to go back to U of T in order to finish my PhD. So I'm actually right now no longer officially a part of Deep Genomics. So also want to make it clear that any of the opinions that I'm presenting here are my own. They're not Deep Genomics. But I want to start a little bit talking about the environment that we're finding ourselves in the time when we started the company. Pretty much starting 2012, there was really what I call a Cambrian explosion of deep learning. One of the major contributors to that was this paper that came out from Jeffrey Hinton's group at U of T, where they, for the first time, really beat the common computer vision algorithms at the time using a deep learning method and improved on the. So they had about 30% relative reduction error, where the field had been pretty stagnant for years before that. And just a couple months afterwards, those guys founded a startup as well, and were immediately bought out by Google, which kind of kicked off this whole talent wars in the AI space by the big tech giants. But we were working in biology, and still this was really relevant to us. And as deep learning went on to touch so many other domains like natural language processing, machine translation, speech recognition, and so on, it was really becoming clear that it was going to be a force in biology as well. But so why do we really need AI or deep learning in genomics? So kind of like a stereotypical problem that we have in biology is that we have what's called a genotype. That's the content of your genome, and somehow this genotype produces a phenotype, which is some biologically determined trait. It can be really high-level stuff like the color of your eyes, or your height, or whether you're lactose intolerant. But in genomics research, we're very often interested in very low-level phenotypes that have to do with gene expression or splicing, so things that you can measure at the molecular level. And a lot of the facts how these things are regulated are known, but they're kind of known as isolated facts. So you don't really know how they interact. And to understand the system as a whole, then if you want to do that, then it really becomes necessary to use tools like machine learning and deep learning. So we were doing research in this space, and in 2015, we had two big research projects that came out from our group. I was involved in the one that's on the left. That was a project that predicted a process that's called alternative splicing. And the project on the right, that were my colleagues, but they were this was the first time ever to apply convolutional neural networks to DNA sequence. And so we had this body of research, and there was kind of like growing interest from the academic community. But to us, it was really evident that these methods were going to be really important in medicine, but we didn't really see a big wave of pharma companies and clinicians coming and beat down our door and wanting to use these tools. So that's what kind of gave us the impetus that we felt maybe it would be a good idea to start a company of our own. And really what was, so I've kind of described the growth in deep learning that was going on and how our tools kept getting better and better. But the parallel development was this astonishing drop in the cost of sequencing. So in 2003, sequencing the first human genome cost about $3 billion. And around 2015, that dropped to below $1,000. So the rate of decrease is actually far faster than Moore's law for that. And when you look at it like this, it was becoming clear to us there was just this massive opportunity opening up because clearly all this genomic data that was being produced would have to be analyzed somehow. And the existing methods were not going to scale to these amounts of data anymore. So starting from that, we developed our business plan, and so the idea was that we would provide automated variant analysis for clinical purposes. Using or providing pre-computed data sets and software as a service that kind of wrapped our predictors that we had built in order to be used by genetic counselors, genetic diagnosticians in order to help diagnose genetic disease in patients. So when we set off and we found several angel investors. A lot of them were other academics, professors who had started companies before and believed in this idea. We joined several incubator programs that were also affiliated to the University of Toronto, and that gave us exposure to a lot of mentors, support for lawyers, and access to cloud computing and lots of other things that were really helpful. And then in October 2015, we went to Silicon Valley and we pitched this to investors, and were eventually found a really great match, and we were funded with $3 million, which really meant we were kind of off to the races, and we found ourselves our office, and we started hiring people, and really working on this idea, developing our predictors, developing ways to deliver those predictors, and we talked to our target audience, to the genetic counselor and the clinicians, and and that was going really well. But nevertheless, in the end, after about 2 years of doing this, we still pivoted because we ran into a couple of roadblocks. So one reason was something called the ACMG guidelines, which are the clinical guidelines for providing this sort of variant analysis. And the way they are written is that they regulate the actual technologies that a clinician is supposed to use to provide the interpretation of a genetic variant. So even if we could demonstrate that our predictors were clearly better, we would still need to work with these regulatory bodies in order to get them approved. But kind of the other thing that we realized was that it was harder than we expected for clinicians and people not trained in machine learning, people who didn't understand the intricacies of the models that we built, to properly interpret the scores and the predictions that we were providing. And so eventually that led to the realization that if we really wanted to maximize the value of the models that we were building, we ourselves were much better positioned than the clients that we had in mind to realize that value. And another development that was going on at the time were that the first drugs of a particular category called antisense oligonucleotide therapies were being approved. And that is a class of drugs that's a kind of genetic therapy, but one characteristic of those is that you can design those drugs much more rationally than the typical small molecule drugs that which most existing drugs are a part of. So those drugs are literally short segments of DNA or RNA that have a therapeutic effect if you introduce them into the cell. So this led to our pivot, and we changed our business model to develop those drug candidates and to build a platform to find and develop those drug candidates more efficiently and more easily than it was being done at the time, and then go to pharma companies and license those drug candidates to them, such that the bigger pharma companies that would have the resources to take them through clinical trials could do that, and so eventually, when those drugs go to market, we could receive a royalty on them. So how do these ASO drugs work? So this is an example of one of the very first ASO drugs that was approved and is actually on the market now. So this was done by a different group and a different company. But this is a drug called Spinraza that is really a game changer for a condition called spinal muscular atrophy. That's a type of muscular atrophy where in the most severe cases, babies born with this condition survive for less than 4 years because they develop almost no motor function. And the problem is that they have a defective version of this SMN gene, which encodes a really crucial protein for muscle development. And the way this drug works is that it's an actual short piece of genetic sequence. You introduce it into the cell, and it's an exact complementary match to a particular position in the genome. And so these blocks that are numbered 1 through 8, those are called exons. Those are the genetic sequences that contain the code for this protein that's being built here. But usually the exon 7 is being skipped, which leads to the protein not being functional. And when you design a short snippet of sequence that cleverly attaches in this location, it will block a genetic signal that causes the skipping of this exon. So the result is that this exon is now being included, which leads to the production of a functional protein, and therefore for the patient developing much better motor function than without. And so this is really a nascent category of drugs, and there's a number of other genetic conditions that this will be applicable to. And so what Deep Genomics is doing now is looking at a number of those conditions that are good targets for this technology. And now we're essentially using our platform to answer questions like in which position exactly should you bind in order to affect the genetic regulation in the way that you want. So lastly, I just want to summarize a little bit what kind of my learnings are about starting a company in this space, and also seeing other academics starting AI companies and either being acquired or going on their own. But for me, the main realization really was that AI just by itself is not a product. So if your predictors and your models may be as great as you want, it's very rarely a sustainable business to just wrap that in some API and putting it somewhere and letting people use that predictor. If you really want to realize the true value of your models on LPI, you really have to do the work of deeply understanding a particular sector and being very careful about the applications for AI that you choose. And you still need to, and if you want to develop a company that can stand on its own without needing to be acquired by some bigger corporation, you still need to do all the other aspects that are necessary for your business as well. So Deep Genomics started as a purely computational group. All we were doing were bioinformatics and machine learning, and so now we have a wet lab that may ultimately become bigger than the computational group in the company. We're doing software engineering. We're building our own tools for our internal use. So we thought we had found something that was easy, but it turned out there was really a whole lot more that we had to do. yeah, that's really my realization from this from building this company for the last 2.5 years. And thanks for the attention.
Great, thank you. Do we have any questions?
Yes.
We're going to give you a mic.
Hi.
So I presume that this was a US-bound company?
So we're in Canada.
Okay, so
Yes.
Canada. Well, here on the other side of the pond, we have, well, right now it's kicking in GDPR. So, you know, that has some kind of implications. So I was wondering, how do you actually get your data genome? Okay, the sequences, you know, you're saying that basically you are probably outsourcing that. It's cheap these days. You basically import that in and then you do something with it. As I understand, so you found that particular monetization factor, which was the spin, right? But are you actually storing it somewhere?
So we work with data from a few different sources. So now that we have the wet lab, it's actually our own wet lab that produces the data. And the data is all from cell lines, like from a test tube, so there's no privacy issues around that. Other sources of data are just public sequence databases that have some regulations around them as well. So sometimes you're required to encrypt the data when you store it. But yes, so the way that we built this, we always avoided having to do with actual patient data. And yeah, if we had done that, there would have been regulations in Canada and the US as well. So there's a set of regulations called HIPAA. Yeah, so we didn't actually have that problem.
Okay. Okay, fair enough. At the beginning, you said that you had, I mean, angel investors was what, $3 million, I think, that you got, and that you use it for lawyers. I guess that most of that money actually went to that research, right? If that's even plausible to run a business like this at the time. I mean, this was what, 2015, right? I guess.
Yeah.
So, I mean, was it like that? Was the legal aspect of the thing? I'm not really sure what's the difference between the US and Canada. I think in the US it's a little bit more stringent, but I think it was very clear that you want to expand to the South State, right?
So yes, I think it's shocking for everybody who has no idea about lawyers and gets into this and then learns how expensive they are. So it was not a majority of our funding that went to this. And it's also most startup lawyers, they often have somewhat favorable conditions where essentially they give you a loan. They work more cheaply in the beginning, and then once you get your first funding, etc., then you have to pay them back. But on the other hand, it can be much more expensive not to spend your money on lawyers, because it's with all these contracts, shareholder agreements, etc. It needs to be set up in a way that is watertight. But yeah, it's pretty shocking how much involvement of lawyers you need.
Okay, that's good to know for everybody. So maybe last question.
I'm sorry, two questions already.
Yeah. Oh, so
wait, there are more questions.
Okay, there you go.
Let's take it with last. I'm sorry, I don't know it. I was actually you were touching upon the data question. I think a lot of people would be curious to know more about the data and how do you talk about variations and your response was that you do some kind of you work with cell cultures and whatnot. Could you talk more about how to do deep learning you need loads of data? What are the issues that and insights you gain there?
Yeah, so it is not 100% deep learning that we do. So in certain other areas, we also use more simple models, and yeah, so it is expensive to generate data in the wet lab, and it'll take a while to ramp up to the amounts of data where you can seriously do deep learning. So we're looking at some solutions to that, like for example active learning, because now we have the data generation process under our control as well. So you can kind of see it as like an exploitation trade-off. So we want to screen for the ASOs that have the greatest effect, but we also want to look in particular areas of the design space that we still know little about. But then, as I mentioned, a lot of the deep learning that we've done also depends on public data sets that are theoretically available to everyone, but you end up like putting in so much work into these data sets to extract the kind of signals that you're interested in. It's almost like you created the data set from scratch in a way.
Okay.
Yeah.
How do you get antisense oligonucleotides into the cell?
So that is essentially chemistry. That is not my area of expertise.
So
but
can you repeat the question? I can't hear.
So the question is how you get these antisense oligonucleotides into the cell.
Okay.
Yeah, so this is another area of studies. It's also
I
a
can
startup.
quickly answer the question. You do a transfection with a chemical agent which perforates the cell and inserts the DNA.
Yes, so if you want the short answer, it's there. So the long answer is it depends on the cell type. There are particular chemistries that are being designed, for example, for delivery to the liver. The example that I showed, Spinraza, it needs to be injected into the spinal cord because of the blood-brain barrier that you otherwise wouldn't be able to cross. But yeah, it's essentially it's chemistry and the question of where you inject it. So the other another tissue where you can apply to is retina. So in that case, you have to inject it into the eyeball.
Okay, do we have maybe a last question?
Yeah, how big is the full genome and how do you fit it into the working memory? Do you have some tips or about the right dimensions of that?
So the question was how big the genome is?
Yes. Getting into working.
Yeah. So the whole genome is about 5 billion letters. But you don't work with the whole genome at the time. So you look at particular areas of the genome, like the exons and introns that I showed, and you kind of extract events around that. But yeah, then it really depends on your application, and you'll need to do data management. Model or data parallelism, the same way that you do, in other deep learning problems.
Okay, so maybe I can take the last question. You said that, that you think that only selling an AI model is not a sustainable business. And if I understand it correctly, you think that in the business needs to be vertical. That is, you need to have both the AI model and then the expertise in the field. Do you see any potential for broadening of this? Some kind of, I don't know, umbrella companies or the horizontal part of it?
Yeah, so I think there's actually a few rare counterexamples to my thesis. There's one company. I'm just blanking on the name, but they're building an API for image analysis. And the big Google Cloud, you can just give them loads of speech data and get it transcribed. But you can really see that, you know, the big tech giants, they're really commoditizing that aspect of AI. And then the other area you can go in is consulting. And I think, well, I'm sure that is going to be a big area, but I think it's difficult to enter for small startups. Because, so first of all, I think a lot of value when you're running a consulting company is doing your own engineering and building your own platform, which may be, depending on how ambitious that is, maybe very expensive. So you need a certain scale to do that. But then, obviously, when you're consulting, you're only capturing a small sliver of the overall value that's being generated. So I have a feeling to be a very successful consulting company, you need a certain scale. There's, for example, Element AI, which is also a Canadian company that is going into consulting in a really big way. They've only existed for a year, and I think they raised about $120 million. And so, according to them, they need that much money in order to be taken seriously by, you know, big enterprises that really want to roll this out on a big scale.
I see. Okay, well, thank you so much. And right now, it's time for a 15-minute break. And after that, we'll have a talk by Max Gordon. So grab some beer and so on. And thank you for